I'm happy to announce that you made it happen. DNA Today has been nominated in the 2020 Podcast Awards for the Best Science in Medicine show. This is our fourth year being nominated, and we are up alongside big podcasts like Gimlet Media's Science Versus. So huge thanks to all the loyal listeners that made this happen. If you did nominate DNA Today, check your email to see if you are one of the lucky few with the power to vote for the winner. You can also go to podcastawards.com and click on the blue box in the middle of the page that reads Voting Open. There you can sign in and it will tell you if you are one of the powerful people that can vote for the winner of this category among the others. So thanks again, everyone. Really helps the visibility of the show and having the show grow. So thank you so much.
How is it that we find ourselves surrounded by such complexity, such elegance?
The genes of you and me, the genes of you and me, we're all made of DNA. We're all made of the same chemical DNA. We're all made of DNA.
Hello, you're listening to DNA Today, a genetics podcast and radio show. I'm your host, Kira Deneen. On this show, we explore genetics' impact on our health through conversations with leaders in genetics. These are fellow genetic counselors, researchers, doctors, patient advocates, and many more science communicators. My guests today are from Fulgent Genetics, who's the sponsor of our ongoing DTC series with their Picture Genetics kits. This is the third installment of the series, so definitely go back and listen to our two previous episodes if you haven't already. So my two guests today are Jessica Shiles, who is a genetic counselor, and Dr. Samuel Strom, who is a geneticist. Jessica serves as Fulgent's clinical genetics marketing specialist. So her main role is to provide clinical training and expertise to help develop marketing material that's used to educate, support, and inform the patients and providers that Fulgent serves. With Jessica's strong passion for patient advocacy, she also leads Fulgent's community outreach. She's a fellow Sarah Lawrence College alumni, where she also received her Master's of Science in Human Genetics. Previously, Sam was an assistant professor at the UCLA David Geffen School of Medicine, where he pioneered interpreting genomics results in the context of clinical diagnostic testing for both rare inherited diseases and cancer. At Fulgent, he is continuing to forward this new science at an industrial scale. He's also an accomplished researcher in the fields of neurogenetics, ophthalmology genetics, and molecular diagnostics, with publications in top journals such as Science, JAMA, Genetics in Medicine, and Human Molecular Genetics. Thank you so much, Sam and Jessica, for coming on the show. We're going to be diving into talking about different kits that are provided by Picture, which is powered by Fulgent. Before we do that, I did want to get a little bit of a reference on what we're going to be talking about today with direct consumers and also the other terms that are kind of related to that. Sam, could you start us out by giving us this frame of reference to understand what a direct consumer test is and what the other terms out there are?
Sure. Yeah. Thanks for having us today. So a direct-to-consumer test in sort of its purest form is something where the only interaction is between a consumer or a patient and a company. So something like 23andMe or Ancestry.com, where you go online, you get a kit. Eventually, after giving them a sample, they give you a report, and there's no doctor involved in that process. What we're talking about today with Picture is a provider-supported model where there always is a healthcare provider involved, a physician, and also genetic counseling resources available. And so I think we can kind of dive into what that means and why we're doing it that way. And I think you had a previous episode in this series that went into some detail there. But what we're talking about today is all in that area of healthcare provider-supported testing.
Certainly, and if people are interested, the first episode of this series will really tease that out further. So that's to refresh everyone's memories. So thank you for that. And one aspect within this that I wanted to talk about was just for those that may not understand or are aware of this, what is the difference between genotyping and sequencing? Because we're going to be talking about this during the show. So in case someone doesn't understand this, Sam, could you also explain this concept to our listeners?
Yeah, this is a really important concept, I think. When you talk about genotyping, you're talking about looking for a small number of very specific genetic changes. So use BRCA1 as an example here. So there's one particular mutation in the BRCA1 gene, which is pretty common in certain populations of people. including Ashkenazi Jewish people. Places like 23andMe are able to get their tests verified by the Food and Drug Administration, FDA, for one mutation at a time. So one of these changes. So they have on their test one BRCA1 mutation approved for testing. Whereas when you do sequencing, you can potentially discover any mutation. And there are actually thousands and thousands of mutations in BRCA1, which are known to cause increased risk for breast and ovarian cancer, in addition to that one, which is the most common, but most people who are positive aren't going to have that one. So when you do a genotyping test and it comes back negative, you've only been tested for one thing. It's like going to the grocery store and the person comes out of the grocery store and you say, "Did you get eggs? Yes or no?" And they say no, but their cart is full of stuff. You have no idea what's in the rest of that cart. So sequencing is looking at everything in a given gene that we know how to look for.
That is an interesting analogy I haven't heard before. I might have to use that one in the future and credit you. I think that this is a really important concept for people to understand, that genotyping is limited. If it finds what you are looking for, great. But if it doesn't, that doesn't necessarily mean there isn't something else happening that is affecting the gene and the way it functions. And so I think that's an important concept to understand when we're now talking about the different kits that Picture Genetics provides, powered by Fulgent Genetics here. And one of those is picture parenting. We're going to talk about 3 today. Well, 4 if we're including our COVID-19 test that I'll kind of tease now, that we'll talk about at the end of the show. But for now, the first is picture parenting. So this is carrier screening. Sam, could you explain what carrier screening is and the types of conditions that are being identified through this type of screening?
Yeah, so for carrier screening, you're looking for things where the parents are healthy, but they have sort of this ticking time bomb that can happen in their kids where the kids can have a disease. There are actually a lot of different conditions that behave this way. Some of the most famous ones are cystic fibrosis, the thalassemias. There's excellent conditions like Duchenne muscular dystrophy, where the mother is a carrier and her sons are at very high risk to have, all of her sons would have 50% risk of having the muscular dystrophy disease. And so carrier screening checks parents to see what they have. Ideally, you check both parents and compare them and see if they share any genes that are both mutated, because that's when there's going to be a really strong risk for a kid to have the disease.
And Jessica, bringing in the genetic counseling perspective, is someone that, as genetic counselors, we meet with patients to discuss this type of testing and see if this fits in with their family planning and where they're at in that process. What is the most ideal time to have this carrier screening done?
Yeah, that's a really good question you ask. And of course, my overall genetic counselor answer will be there's no ideal time that fits everybody. Each patient's journey is definitely different. But this test can definitely be taken before, during, or after pregnancy. And actually, believe it or not, the American College of Medical Genetics states that carrier screening is most ideal before pregnancy because it really does allow and enable couples to learn of their reproductive risks and really kind of consider the whole picture to get a complete range of what their different reproductive options are. And this really does kind of align with Picture's values too, just in helping individuals really be proactive about their health and their family's health.
So there isn't too early of a time to get carrier testing carrier screening. From my understanding, I did one of the picture parenting kits to get an understanding of mine. I'm not trying to conceive at the moment. That's far off for me. But it was interesting to see what conditions I may be a carrier for and learning that through doing it. The next kit that you have is Picture Newborn. So we're kind of going along with the life cycle here of now when someone is actually parents and giving birth and having a brand new baby. What is this testing exactly? Is this for sick babies or for healthy babies?
Yeah, it's a great question as well. So Picture Newborn is really geared towards healthy babies. You're likely to think if there's a significantly ill baby and early on in pregnancy, those will probably be identified with newborn screening, and they really do need that immediate care. And what Picture Newborn is there to kind of help with complementing things like newborn screening would be with babies that might seem healthy at birth, but might have underlying health conditions, that might appear within first couple of days or a few months of life. And, a good example of that would be like MCAD or any of the fatty acid oxidation disorders. It's a metabolic condition, might not be picked up right away, but after periods of maybe illness or fasting, these kind of conditions can arise. And if you're able to catch that and have a known risk before, you can kind of change the outcome of these potential symptoms that could be caused by having this condition.
So in a sense, it's almost an extra step to newborn screening. Most babies have newborn screening. It's an opt-out process in most of the states. That's going to differ a little bit state by state. But this is kind of giving extra information because, as you said, with the newborn screening, that's more looking at, it's a dried blood spot that you take from the prick on the baby's heel. So you're looking at more biochemical different proteins and that sense. Whereas this test, we're talking about genetic tests on the show today. So we're looking more to see if the baby has a certain condition or has a genetic marker for that.
Yeah, absolutely. We like to kind of look at it as like a safety net. Not all the time is newborn screening going to pick up these particular metabolites on the blood spots and is not fully diagnostic. So by going through and doing full gene sequencing and looking at the baby's actual DNA is more of a diagnostic confirmation that this child will develop or has an increased risk of developing these particular disorders.
I think it's important to remember that, or just to recognize that what the state does with metabolic newborn screening is maybe the most successful public health initiative ever. It's really been an amazing change. Diseases like phenylketonuria used to cause severe intellectual disability throughout the lifetime, and newborn screening of the metabolites, like you talked about with the heel prick, the key reason that works is because it's something that you can do on a large number of people. You can pick up the people who might be affected, and the key there is that there's something you can do about it. With PKU, you avoid certain foods, a lot of foods, but they will live to be completely healthy in terms of their metabolic disease. So what we did with the new picture newborn is to try to find the conditions that would be tested for by the state if there was something in that heel prick that they could test for. So meeting all the same kinds of standards about medical actionability and screening efficacy. Basically, we started in a room with a conference room with a whiteboard of how do you pick a gene for newborn screening, and just removed the last thing, which is there's a metabolite that we can test for, and changed it to there's a gene we can test for. And that's how we kind of came up with this list. So one example is immunodeficiencies. There's one immunodeficiency test that's done for some kids in some states, but then there's about 24 different genetic ones that you can test for on the same specimen. And it's the same type of disease. It's the exact same reasoning that you'd want to test for it in a healthy newborn. So I think it's a really exciting new way of thinking about this. We think eventually all kids should have this. And right now it's really just getting our toes in the water as a supplement to the state newborn screening. We don't imagine it replacing newborn screening because newborn screening as currently conceived is so effective. There's no reason to stop doing it, but this can sort of act on top of it.
It's interesting that you fill in the gaps where newborn screening can't quite go. As you said, with newborn screening, you have to have certain markers, really, to be able to say, okay, we found this, and this is elevating this newborn's risk for having a certain condition. And then they go on to do further testing because it's newborn screening. We're just screening newborns. Now, with yours, you're talking about it being more of a diagnostic test and able to test for other conditions. So kind of between the two, you're able to cover quite a few conditions. And now linking this back to the carrier screening that we were talking about at the beginning of the show, if a couple had negative carrier screening, is doing a newborn test repetitive? Is it the same conditions that are on your carrier screening as would come up on the newborn testing here?
So it's not the same, but there's some overlap. Maybe we should make a Venn diagram we can put on your blog. There's definitely some overlap. Awesome. Yeah. The key difference with the newborn ones are some of these genes are ones that are autosomal dominant, or even X-linked dominant, or even X-linked recessive. They're ones where the mutation could be a new mutation occurring that's only in the baby. And so if you test the parents, they're going to be negative. But there could be a new mutation, what we call de novo, in the baby, but there's really no other way to test for that right now. And so there are quite a number of conditions on our newborn. We call it newborn genetic analysis. We avoid screening because, as you talked about, it's not really screening. It's a diagnostic test. So there are a number of genes on here that are dominant, where the child would have the mutation and the parents would be negative.
And that's a really good point, that a lot of these conditions, when we talk about genetics, I think people often think of, being passed down through families, but a lot of genetic conditions are de novo, so kind of randomly happen. And so there's a mistake somewhere, and that's not going to show up in the parents. If you're screening the parents, that's not going to end up giving a diagnosis to a baby that is affected by one of these conditions. What I'm also learning with this is that you're only including early onset conditions. Jessica, why is this?
Yeah, I think that's just kind of going back to what Sam was saying earlier with why we picked, some of these genes to be included. A lot of the times, these conditions are occurring earlier on, pretty much under the age of 10. And we really believe that knowing these risks ahead of time can really help parents prepare for their child's life and the quality of their living. So early identification for these disorders can help with early prevention and management. It really makes, the parents feel empowered. They have options. They know that they're really doing everything they can to protect the future health of their children. So that I would say is one of the main reasons why we have these early onset conditions.
And then I think the genetic counseling term we like to use is autonomy, right? So if there's something like BRCA, for example, that's going to be an adult onset condition where there's no risk until that person is over the age of 18, well, let's just wait until that person's 18, and then they can decide whether or not they want to be tested. So that's another thing that was on our whiteboard at the beginning was all the medical ethics. We're not in this to make a quick buck. We want to use the technology to improve health. And in order to improve health, you have to be careful not to make things worse. First do no harm. And so doing adult neurological conditions like something like Parkinson's disease or Alzheimer's dementia on a baby, I think is just wildly, just not appropriate, and it is more likely to do harm to that individual's psyche than to actually help them.
And as you said, you want to allow people to make that decision for themselves if it's not a condition that's going to affect them under the age of 18. So if it's not a pediatric condition or going to have an onset or have symptoms while they're still a child, then usually it's something that we like to save until someone is an adult and can make the decisions on their own. And that leads into your other kit, which is Picture Wellness, which is looking at a certain number of conditions that kind of fall more into this category of being affected, when you're 18 or older. This is based on the ACMG 59. Sam, can you explain what that even means for people that have never heard this before?
Yeah, for sure. So the American College of Medical Genetics and Genomics, they've added a G, but everybody kind of forgets that that's a thing because ACMGG just sounds like you made a mistake. But that's the medical profession, the professional society that is in charge of licensing genetics professionals, specifically medical geneticists and laboratory geneticists like myself. And so that group came out with a list of originally 57 genes that their committee had decided sort of met several criteria. First of all, that they were high risk, so that if you have a mutation in one of those genes, you're very likely to have the condition. That again, the idea of medical actionability, that there's something you can do about it, that there's for cancer, you can do increased surveillance. For a lot of these are cardiac. We'll talk about sort of the diseases in particular, but you can do things like maybe install a pacemaker before someone dies of a heart attack. So that's sort of the list is these genes where they're well established. the genetics community is very confident in these genes, and there's medical actionability surrounding it. The list we're using now, the 59, is actually version 2.0, so they published that list, and then got a lot of feedback from people around the community around the world, genetics professionals saying, hey, you forgot about this gene, or hey, this actually one of these genes we're not so sure about. And so they published a sort of an updated version called the Secondary Findings 2.0 list of 59 genes. And that's we basically developed a laboratory test that can evaluate exactly those 59 genes.
And so this is not a choice that just two people sat down and were like, oh, let's do these 59. This has really been in the genetics community refined and discussed and debated to see, okay, what are these final 59 genes? At least for now. I'm sure there will be, in the future with more research, this will be reevaluated. But if someone is healthy, Jess, why would they order this test? If they are healthy themselves, maybe they have a good family history, why would someone want to order Picture Wellness?
Right. Yeah. I mean, I think we're in a culture where we really just want to, kind of be in control of our health, and anything that we can really kind of get our hands on that can help with prevention, management, screening to do that. We're likely going to kind of jump onto. And I mean, there's so many complexities and uncertainty in our health. And if we have the opportunity to be proactive and take a Picture Wellness test to kind of figure these things out and learn our risk ahead of time, really, I keep coming back to this term, but just feeling empowered and being able to be educated about yourself and your health, and being able to take this information. And like Sam said, especially with the ACMG 59, these things are clinically actionable. We can actually put action to these types of results. So, I mean, to each their own, but most of the time we really like to kind of have control of our health. And if we're able to be proactive about it, I would say that this is probably the reason why people want to and would order this type of testing.
And one thing I kind of meant to talk about and didn't, about how this list came about. It originally was not sort of in the context of testing healthy individuals. It was actually in the context of testing people who are already getting a genetic test because they have a genetic disease or a suspected genetic disease. But you're doing something like an exome or a genome sequence where you have data for 20,000 genes to do a whole exome test. There was a question about, well, what if you found a mutation in one of these genes in someone who you're testing for something else? And so that's why it's called sort of a secondary or incidental findings list. The need to come up with a list sort of sprung from that. It was, okay, so we've tested this person. We're coming back to BRCA a million times, but I think it's a good one because people are familiar with it. What some people may not be familiar with is that people who have both copies of their BRCA gene mutated can have something called Fanconi anemia, which is a severe pediatric condition. It has skeletal abnormalities and abnormalities of the blood. So you might have a kid who has what looks like Fanconi anemia, and you're doing an exome. You can find the genetic cause of their Fanconi anemia. You may also find a mutation, that might be one of the other Fanconi anemia genes, but you also find a BRCA2 mutation that means they're going to have increased for ovarian and breast cancer later in their life. So there's a lot of complexity. And so the ACMG wanted to come out with a list to say, like, if you're going to do an exome sequence, originally they said you must test for all of these, whether the person wants it or not. And there was a lot of feedback about that to say, well, people should be given the choice to opt out. If it's a child with a severe, neurological condition, maybe we don't really need to know if they have risk for breast cancer. So the idea to opt in or opt out kind of came about. But those conversations always kind of turn back towards, well, if you're going to test somebody with, a genetic disease that's unrelated to these genes, and you're saying that that's medically necessary, what about everyone else? Why? Are we testing the parents of those people? Oftentimes we are testing the parents of those people. Do we tell them about their results? So it got, there's sort of this like unraveling of the sweater that kind of happened where it's like, well, wait a second, why aren't we doing population screening on everyone? And I think, as Jess was saying, we're in a society where many people would like to have this information, even if they don't have a positive personal or family history. And it can be very difficult for them to get a test right now. If you want to know your status for BRCA, if you don't have a history, until recently, that would be a very, very expensive test that, maybe if your physician wouldn't do it for you. And so the picture wellness really gives the power to the individual to say, well, I'm interested in knowing this information. The technology is there. We can do it. And can we do it in a safe way that's affordable? And so I think we're sort of threading that needle.
I think it's important for people to have this option. As you said, when they're getting, say, whole genome or whole exome sequencing, I've been with patients and they're learning more about this and saying, okay, am I opting in to get this information or not? And having that be an option. And this is one area of genetics that isn't necessarily in all areas where there is a consensus on this of what these conditions are because they're actionable, that patients can do something about them, and that it is advantageous for them to learn about it if they so choose. So I think that's interesting that the genetics community has been able to come together for these conditions, and will be interesting to see if other conditions are added to the list and at what point they are. But before we end the show, I did want to talk about one more kit you guys offer, and that is a recent kit due to the pandemic, the COVID-19. Sam, how and when did Fulgent start doing COVID testing?
Yeah, so back in February, my boss of 2020, my boss started saying, we're working on developing a test for COVID, for this virus. And if you remember, this is sort of before it was a big deal in the United States. It had first occurred in China, and then there were some other countries, Italy and Spain, and I was kind of shocked because in my training I did some work with infectious diseases when I was a student, but it's been a while, and it's not something that I was used to doing. And so, it really became our number one project to develop a COVID test. And I got to say, it's just been amazing to be a part of this team to come together like this so quickly, where we went from not doing any infectious disease testing, not even really having the right equipment, to having a fully operational lab. We're now serving Los Angeles County and our local neighborhoods, and also many other states. So we've been running diagnostic testing for COVID since late March of 2020, bringing it into sort of onto the picture platform was something we thought about early on and wanted to make sure that we did it right, because hopefully people are able to go out and get testing. There's a lot of like drive-throughs and stuff where you can do it in different parts of the country, but it's not like broadly available to anyone who wants it in every place. And so we brought in the same type of approach we brought to picture wellness and picture parenting and picture newborn, to be able to offer COVID testing. So it's a different collection method. The nasopharyngeal swab, we have a video and stuff. You stick this thing up your nose instead of in your mouth. Otherwise, it's actually kind of similar. How you go online and order the test. I think Jess probably could do a better job explaining how that process works.
Yeah. So, I mean, it is, like Sam said, it's similar to the platform that we have set up for Picture, where, you're able to order this test online, at home, safe in your home, especially with the risk of infection being really high where it's at. So this is in the safety of your home. And not only that, but it is mediated by a physician. So when you hop on our website at picturegenetics.com/COVID-19, you are able to order the test. You take a quick eligibility questionnaire that's set out by the CDC. It's approved by the third-party physician company that we're working with. Portal is created. Everything is then mailed to you. You collect your sample, you mail it back to us. And as Sam mentioned, there's a video with how to take the nasopharyngeal swab. There's written instructions provided as well. And then your results are available within your portal within 1 to 2 days of after you send in your sample. And then following that, again, with this whole concept of we really like at Picture to be able to help guide and support you throughout the entire process. You're able to call and schedule a consultation following your results, regardless of whether or not they're positive or negative. And that goes to all of our Picture product lines, with the genetic counseling support at the end. I think is one of the biggest, again, differentiators between the direct-to-consumer testing versus the physician and healthcare mediated. We really want to provide that support at the end. If you have follow-up questions, again, whether or not your results are negative or they're positive, to be able to provide and shed some light and insight onto those things. Those individuals are there to help you when you would like.
Well, thank you so much for offering the COVID test, because there are people that may not be able to drive out and get tested in person. So being able to have a kit sent to their house, I'm sure, is just great for so many people. And it really is very quick and easy to order one. I think I ordered one in maybe 4 minutes, 3 minutes, something like that. So it was very quick, and just really appreciate you guys stepping up during the pandemic to offer your part. As Sam had mentioned, you hadn't previously done infectious diseases testing, so this was a new front for you guys, and kudos to you for being able to offer this test. And thank you in general for coming on to this episode. We explored a lot of kits that you offer and just the concepts behind them and what people should be thinking about and understanding when it comes to these kits. And that at the end of the day, you also offer genetic counseling with all of your kits. So even if they're not soaking everything in during this episode, that's something that they can get personalized genetic counseling. So thanks so much. This has been a treat to explore all this with you guys.
Yeah, thank you.
It's been a pleasure. Thanks, Kara.
If you are interested in getting your own kit, we're doing a giveaway. So head over to our social media by searching DNA Today on Twitter, Instagram, and Facebook to enter to win your own free Picture Genetics kit. And if you can't wait to find out if you have one or you want multiple kits, head over to picturegenetics.com with promo code DNA Today for 25% off and free shipping. Again, the code is DNA Today for that 25% off and free shipping at picturegenetics.com. And all the links we've mentioned today throughout the episode, other episodes, they're all at dnapodcast.com. So you go there, all the information you need is there, including the contact form on our website. If you have questions, you want to directly email in at info@dnapodcast.com. You can send questions to Jessica, Sam. I will forward those on. And thank you again for everyone that helped to get DNA Today nominated in the podcast awards. If you're one of those people, don't forget to check your email to see if you're one of the lucky few that has the power to vote for the winner. So we've been nominated, but still up for grabs of who is going to win the award. Now, here's how it works. Through random selection, 5,000 listeners were selected to be able to vote. So if you're one of these people, it's your job to help get the show more votes and hopefully a win for us. So if you're not sure if you're one of those people, head over to podcastawards.com. You can click that blue box in the middle of the page that reads voting open and sign in, and it will tell you if you are eligible or not. So again, big thank you. Another way that you can help the show receive more visibility is giving us a rating and review on iTunes, or now we call it Apple Podcasts. So definitely be sure to do that if you haven't already. Thanks for listening, and join me next time to learn and discover new advances in the world of genetics.
The genes of you and me, genes of you, and they're all made of DNA. We're all made of the same chemical DNA.